scoliosis being a curvature of the spine. Remember, kyphosis is the upper thoracic spine bending over, usually from compression fractures in the front and the anterior bodies. Lordosis is that abnormal curvature of the lumbar spine. Scoliosis is sort of a twist. And here's a nasty one. And usually it's mostly a thoracic spine problem here, but you can see how the ribs are going to be abnormal, and you're going to have a real, twisting of the thoracic spine, especially when you bend over. You're going to see a real difference in the height when you see that. They usually measure the angles. They take a line from the intervertebral disc and draw a line, and then they draw it up this way. And the wider that angle is, the worse off the curvature is, obviously. This is the kind of thing. Here's a 57-degree curvature on this one. Okay, and after the repair, they put basically bars and pins in there and straighten it up. And they got a 32-degree curvature afterwards, which is much better. But usually you're not going to get them perfect. Okay, because you really got to push these things around. So it's more like, I always say that orthopedics is more like carpentry than anything else, because it's all nails and screws and pins and everything. And they get in there with hammers and all sorts of stuff to straighten these things up. But, so that's scoliosis. Here's what you're looking for in kids. Okay, you see that little lump in the curvature. It may not be quite that pronounced, but and you can't always really see. Sometimes, actually, the curvature, the anterior bodies can spin over, but the spinous processes may remain in a straight line sometimes, but you can still curve the front over and get that scoliosis. So you're not always going to see a line that's abnormal. Here's an unusual sort of situation. I've seen a few of them, maybe 10 cases in 20 years. Okay? But you'll see this is a rib coming off the 7th cervical vertebrae, and occasionally it causes swallowing problems or problems with the voice. I know of at least one opera singer who had it. She had it removed because they were affecting her voice. They're coming up in the throat too closely. Occasionally it causes problems when it stretches the cervical plexus up here. These nerves are coming down and going over that first rib. And if you have a high rib up here, it stretches those nerves a little bit. And occasionally you'll actually have a rib that will come around and come over the top of the nerves, and it will actually pinch in between the two ribs. Okay? So whenever you move your arm, it starts to go numb or tingly, and that's when you get symptoms, when you lift up your arm and when you move it like that. So that's a cervical rib. This is osteoporosis. You've seen what that trabecular bone, that spongy bone, looks like, especially on the tibias that are all beaten up. You can see that trabecular bone on the top of it. Well, that's what the inside of your vertebral bodies look like. And when that thins out, look at this. This is why they crush. And this is that kyphosis I was telling you about. It's because the anterior vertebrae here, they've been crushed. Okay, and they're not square anymore. They're starting to angle in, and that's what gives the whole spine an abnormal curvature. Occasionally it'll get like this, where you can't even see the bone, it's so thin, okay, down in here. So it should be pretty white like this, but when you start seeing really dark bones like that's osteoporosis is pretty bad. It's probably what my mother-in-law's look like. I haven't seen her x-rays, but she's got a little issue with that. She broke a rib just moving some furniture. So some bones are pretty thin. This is what a normal lumbar spine x-ray looks like. I'll show you this because you're never going to see bones again in the hospital. You're going to see x-rays, you're going to see ultrasounds, and MRIs and CAT scans. So this is what you should be kind of familiar with looking at, really, if you want to study the anatomy. But you'll notice here we have the nice disc space here. Okay, all the vertebral bodies are all lined up. And even the promontory on the sacrum here is lined up with the 5th lumbar vertebrae there. Okay, so there's no slippage. But this is all kind of hazy back in here. Okay, this is a straight lateral film. What you also want to get is an oblique film, where they actually turn the patient a little bit and then take a shot, and then this is what you get over here. And now you can see those superior articular facets and the inferior facets. You can see the joints. Okay, and this is what you're looking at a lot to see what these articular facets look like. The eye here, that's the transverse process sticking out at you. Okay, slightly at an angle. These would be the spinous processes back here. But you'll notice that they call this the Scotty dog. Here's the ears, here's the face, there's the eye, here's the leg, here's the rear leg, and then there's the tail. So it looks like a little dog. And the radiologist will actually say on his report that Scotty dogs appear normal. So, but you can get a much better view of these joints at an angle than you can when you're looking at it like this. Here's some problems that can go wrong. You can have the inferior articular processes, it's especially common on L4 and L5 with low back injuries, where you can fracture those off, and then you've got nothing holding L5 from slipping forward. Okay, that's called spondylolysis. If you're born with it, they call it spondylolisthesis. You can actually be born without those connections anywhere along here, and then the spine is unstable and it will slide forward. Usually L4, L5. Usually you'll first diagnose this in a teenager when they start getting more active or doing rough sports or football or something like that. They'll start to have back pain, and you just get a routine X-ray and you see, my gosh, that L5 has slipped way down on the sacrum there. This is pretty normal here. This is the annulus, the fibrocartilage I was telling you about, and the inside is a little softer material. If that ruptures, it can go back and put pressure on the spinal cord, or it can come back here and put pressure on the intervertebral foramen here, where the nerves are coming out. Okay? So that's when you get symptoms running down the leg and that sort of thing. You can actually be born with a stenotic canal where it's too narrow and it starts to put pressure on the spine. My neighbor actually has had a couple of operations for this. And so, or occasionally just arthritis. The arthritis in these areas that can start to calcify and cause a stenotic canal as well, and start pinching the nerves. This is the kind of thing you'll see when they're born without the articular processes here that hold the vertebrae in place. So you'll see, you'll draw the line coming down, and then you got this big drop off here. Okay, it should be coming down straight, level with the promontory. Okay, so here's one on L5, here's one on L4. Okay, and that's the kind of thing you're looking for, the abnormal drop off there. Here's one. Okay, see how L4 is flipped way out there. Okay. Here's a herniated disc on an MRI. These are the discs, these are the vertebrae, and you can see the dark bulge back here. This is probably, that's sacrum, okay, that's L4, L5. So I believe that's that low. Usually the cord, so here's the spinal cord. The cord will usually stop around L1 or L2, so this is fairly low. It's more like L3 here. But this is why you do spinal taps coming in down this level. So if you hit anything, you'll hit one of these nerves that's running down the canal, okay? But you won't hit the spinal cord, okay? This is tuberculosis. Look at his spine up here. You're going to see patients, and then you're going to see X-rays. You're not going to see bones. But tuberculosis, in case you don't know, is a bacterial infection. Get it in the lungs. It can be a chronic thing that destroys the lungs over years. But it also can get in your blood system, and it can take up residence in any of your bones. It's common to get in the femur and in the hip, in the pelvis and in the spine. It's also common to get in lymph nodes and cause them to grow and grow. And you get big lumps in the neck from tuberculous lymph nodes. That used to be a disease that they called scrofula. It was so common. It was real common in like in the Appalachians, out in the wilds and the country and that sort of thing. But you don't see it too much anymore because they treat people for tuberculosis now. But this came from unpasteurized milk. Cows carry tuberculosis fairly often, and the reason that Pasteur was pasteurizing milk was to stop tuberculosis. Okay, it wasn't just to keep it fresh and keep it from fermenting. He was trying to figure out a way to kill the bacteria in there, because a lot of people in Europe were getting tuberculosis from milk. You only see this in areas, this happened to be a shot I found online from Mexico. They get about a dozen cases a year of this down there because people are drinking unpasteurized milk. And you're starting to see a little bit of it in this country because people are starting to think, wow, it's organic, I'm going to drink unpasteurized milk. Boom, there you go. Okay. But the bacteria has just eaten up the vertebrae and they just crushed. Okay. So he's basically got a broken back there. Okay. You can see the vertebrae there, and this one is just totally comminuted, just fractured. Okay. This is something that's a little unusual. You see it occasionally. I've seen a couple of cases, but it's a rare sort of thing. Ankylosing spondylitis. It's an inflammatory disorder, almost like rheumatoid arthritis, where the joints, you have antibodies and the joints start eating away at themselves and destroying the bone. Well, this is a disorder that destroys the bone, and you can see how the vertebrae here are misshapen. Okay, and it's usually in younger women that you first notice it. But the spine just sort of falls apart, and they have to take really strong anti-inflammatories. See how this one's eaten up here? This is a side shot. Okay. You can see how MRIs are really good at looking at some of these things. So, here's a traumatic injury. This is a broken back. Okay. Can you guess what level that is? What vertebrae is that?
L5? No.
L1, L3.
Well, does L3 have a rib?
No.
Okay, and it's a thoracic vertebrae. So is this one. That's the last rib. So this would be L1. This would be T12, T11. Okay, so it's a fracture between T11 and T12. This person is paraplegic. You can't get that kind of fracture. The cord doesn't stretch over. You know, just you cut through it. Sure. So here's a normal cervical spine. Again, you see a sort of a normal curvature here. Okay, when this is really straight, I was telling you that's when they talk about whiplash, muscle spasm. But when you lose this curvature, usually you've got some neck injury, neck problems. The vertebral spaces are fine. The intervertebral spaces are fine. The open intervertebral foramen, they're all open. Okay. Here's the axis of its little spine there. And it should go 1, 2, 3, 4, 5, 6, 7. It's very often, see all this white stuff, that's the shoulder, soft tissue in the shoulder. If you don't pull the shoulders down, very often it hides C7. And many doctors have been sued because they didn't check C7 because they couldn't see it, and that was where the fracture was. Okay, so you got to pull the shoulders down and get C7. If you don't, you're going to go right back to the X-ray department and get another one. So, but these are nice, all in line, not too much arthritis here in the joints. Okay. Now here's one that's not normal. You can see the endotracheal tube in their lung. Okay, so this is probably either an intensive care or an ER shot from a cervical fracture here. You can see how these are all nice and lined up here. This is the superior articular surface. This is the inferior. Here's the inferior. Here's the superior. But here's the superior and here's the inferior. Okay? This one right here should be back on this top of this thing. Okay, and that's why you're getting this big drop off there. Okay, this thing, it's not only fractured, you can see the pieces where it's broken off, but it's dislocated and shoved forward. Okay, so that is probably a quadriplegic. Okay, and you get them up that high, and you're going to have very little motion down below if that's permanent. So, typical neck fracture. This is a big blow up of a regular X-ray of a Dens fracture. You can see the little drop off there. Okay. This is the Dens on the axis, okay, but it's fractured right through there, and there's the edge where it's fractured. And this is the atlas on either side. It takes this kind of special X-ray to get these because you got to take a shot through the mouth. So you get the patient to open their mouth, and you take a shot right through their mouth, and that's why you see the fillings in their teeth right in the corners. Okay, you get the upper and lower fillings. Okay. Here's another one. This is a CT scan, big fracture through the axis. So here's the Dens, okay, and it fractured through the axis and got the whole thing. So this is the kind of thing where you see people with those halos, metal halos to stabilize that. So, this is just something that's not too out of the ordinary, but like it says here, the patient came in with a ruptured appendix. So you see all this air in the abdomen over here. This is just air in the colon. Okay, that's what it looks like. This is more air in the colon. That's air over here. When you get air in the abdomen, though, sometimes the muscles will stand out. This is the psoas major muscle right here. It attaches along the lumbar vertebrae and then runs down and attaches to the lesser trochanter on the femur. And that's the big muscle that pulls your leg up and flexes your leg. That's actually the one where you get filet mignon. That's the steak in the cow. They cut that up. It's a nice round muscle that goes down. But you can see where the fifth lumbar vertebrae has joined with the iliac crest over here, and now this is not movable anymore. Okay? So this is just like a normal variation you see occasionally. Occasionally you'll see 6 lumbar vertebrae, or you'll see 13 thoracic vertebrae. But that's just something that can happen. So, and I was telling you about the transverse foramen, the vertebral artery coming up through the cervical vertebrae, the hole. If it gets pinched off, especially up high up here, or if there's trauma to this area here by jerking a neck or something like that, chiropractors have been sued many times for causing strokes when they jerk a head. And what you do is you tear the vertebral artery and then you stroke out the brainstem. Okay? But this was called a bow hunter stroke because obviously you turn your head to shoot a bow, and so I don't know. People were passing out while they were shooting a bow or something. I don't know, but it's just called a bow hunter stroke. And this is craniocleidodysostosis. This is also an axial problem. This is a genetic disorder where you're born with no clavicles. They look normal like this, except that there's a little depression in the shoulder because they don't have the collarbone. But they can actually take their bone, the head of the humerus, and they can touch each other because there's no clavicle to keep them apart. Okay? So they don't look like this normally. They're just doing this for the picture. Okay? But so they can pull it back, but you'll see the depression. There's no collarbone there. Okay? And somebody was asking me about this, which was strange. One of the students in the class last night said that her friend has this, and I've never seen this in the States. It's a really rare disorder. But this guy, I'm not sure, they almost look Malaysian or something, but the story I heard where it's most common is a fellow who was actually Chinese, but he was Muslim. He immigrated to South Africa in 1898, over 100 years ago. He had 7 or 8 wives, 30-something children, okay? This is a dominant, not a recessive, but a dominant gene, so 50% of your kids are going to have this disorder. There are now over 1,200 people in South Africa, all related to him, that have this disorder. Because it doesn't affect reproduction or anything like that, so it's just going to get more and more common as time goes by. Okay? And, you know, it's interesting to think, you know, if the comet hits and destroys everything but South Africa, sooner or later, all human beings will have no collarbones, because it will just get more and more common until everybody's got it. Okay? So, interesting. They also have usually some facial irregularities and some teeth development problems. Sometimes the skull is, the fontanelles don't close. That's why they call it cranio. Cleido means clavicle, okay? But that's why they call it that. So, here's a shot, no clavicles. Okay. There's a little, looks like there might be a little remnant here, but certainly nothing that's touching the sternum, and it's missing over here, totally. So, this is typical chest x-ray otherwise. You can see the arch of the aorta here as it goes up and then curves down, that goes back around the heart. These are blood vessels and the bigger bronchi that gives these streakiness to the lungs here. Here's your facet, the head of the ribs on the vertebrae. This is your trachea, this dark area here, that's your airway. And then these are the spinous processes in the back that you're seeing. Okay. I told you I'd show you a picture of sinusitis. These are clear funnel sinuses here, ethmoids, fairly clear. This maxillary sinus is pretty clear. Looks like there might be a little swelling right in here. But otherwise, this one is socked in with fluid, and and that's what it looks like on x-ray. If it's not socked in completely, you see the sinus is open, but over here you actually see a fluid level.
Yeah.
Okay, it's just pus just sitting right in the sinus. Okay? But that's what you're looking for when they do the sinus films. And again, since I've had a couple questions about what's the difference between a spinal tap and an epidural, I just thought I'd show you this. Again, your cord usually ends about L1, and then you have the rest of the lumbar and sacral nerves that go on down through the sacrum. So you want to get down here around L4, L5 to do your taps. And you get them to curl up, so that opens up that space between the vertebrae, and then you go down low. And basically you just count the spinous processes going up, and then that's where you figure out where you're going to go in. You go between them, and you'll feel a little snap when you get into the ligament. And there's a space right here. This is the dural membrane here. Okay, this runs all the way up and then runs around the brain, and right underneath that is where the spinal fluid is. And so you want to get into, well, no, sorry, this is the dura here, and the fluid is right below it. This is the ligamentum flavum here. When you pop through that ligament, you can actually feel a little pop when you get through it. And then you know you're in this space. This is where you would put an epidural in this space. Okay? If you want spinal fluid, you've got to go all the way through the dura there, and there's the fluid underneath it. Okay? So it's a little different. You go through the ligament, and then you put the needle and slide the catheter into that epidural space out here. This is where the fluid is underneath this membrane here. So the spinal tap would go here, epidural stays out here, and then they just inject the lidocaine or whatever they're going to put around here, and it just numbs up all these nerves. Okay? So that's why you're not supposed to get any fluid really when you do an epidural. Okay? So, okay. That was enough of that. Any questions on that? This one here, we'll finish up. Guys. Where did we stop?
On the peroxisomes. That's where we talked about that.
Alrighty, we're just working our way through the organelles and peroxisomes. This is a very simplified peroxisome here. Just lifted this picture out of Wikipedia. There's all sorts of membrane proteins in here. There's at least, a dozen or so, at least, oxidative enzymes that will tear apart things using usually oxygen or hydrogen peroxide processes. So they're known for oxidizing things like fatty acids. It's one of the important things they really have to do, other than just detoxifying alcohol, okay, making the aldehyde out of it or the acid out of it. So if you don't have working enzymes, which means any kind of genetic disorder that affects the genes or that make the proteins that carry the proteins into the peroxisome, there are carrier proteins that bond to the membrane here and then bring the active catalyst proteins inside. And they all have their own little separate receptors and all sorts of things. So there's dozens of different kinds of genetic disorders that can affect peroxisomes and make them not work. And especially when you can't metabolize your fatty acids, they start to build up. And that causes, well, anytime you build up something, the tissues that seem to be the most susceptible to damage are nerve and muscle. Okay, so peroxisome disorders, just like the other metabolic disorders, where the storage disorders with the lysosomes, a lot of neurological type problems, a lot of muscular type problems and complaints, and a lot of digestive problems with this too. But the disorders of peroxisomes are usually fatal. A couple of years maybe, usually within 6 months they'll kill the infant. Because they have feeding problems. You got to feed them special diets that don't have these fatty acids in it and stuff like that. And it's a real effort. But if this is catalase, this is a picture of the enzyme that uses, it destroys hydrogen peroxide, but it will also take hydrogen peroxide and oxidize alcohol, okay, and decompose most of the alcohol that you get in your system. Amazing thing. It's hard to believe how fast these things can work. I mean, look at this. It says convert millions of molecules of hydrogen peroxide to water and oxygen per second. So this thing is banging away. Okay, on a molecular level, it's hard to imagine things working that fast. But hydrogen peroxide is HOOH. It's not HOH. That would be water. HOOH. It's an unusual sort of molecule. Well, you've probably seen it in the brown jug that you can get the 3% hydrogen peroxide, and you pour it, and it starts to fizz. It's oxidizing anything it gets its hands on, and it's actually creating oxygen. Okay. We in the lab, we used to use 70% solutions, and these things were really strong oxidizers and almost explosive if you're not careful. So if you can, what I want you to know for the test, well, this is the most important enzyme in the peroxisome. So if you'll associate catalase with peroxisomes, and then remember they're oxidizers, okay, and it's using hydrogen peroxide. But this is also the important organelle that helps your liver detoxify many things in your diet, especially alcohol. Okay, so if you can sort of put those keywords together, you'll get any question I'll ask you about it. Interesting molecule. I don't know where the active site is, but you can see all the beta sheets, and you've got some of the alpha helixes. Okay. And this is all one molecule, so it has tertiary structure, but no quaternary structure that I'm aware of. Okay, so it works by itself. Zellweger syndrome, PBS, just throwing out. Like I said, if you can't digest your long-chain fatty acids, they start to build up and cause toxicity in the cells, especially nerves and things like that. As you can see, this one, death usually occurs by 6 months. And here's a shot of a little kid that has it. They're feeding him because they usually are very lethargic. They don't feed well, they have no appetite. They have an enlarged liver and spleen. Sometimes there's facial abnormalities where they almost look like they have Down syndrome. Sometimes the epicanthal folds of the eyes will move in a little bit. I don't see much of it here, and then it's also known for a high forehead, this particular disorder. So the really good pediatricians can diagnose this just by looking. So people have seen it enough. All right, centrioles. Very high magnification here. Okay. This is sort of a cartoon picture of what those centrioles look like, and when they come together, they form that complex called the centrosome. But just remember centrioles. They really have a set of microtubules like this. Those globulin compounds come together and form that tubule like I showed you before the last test. And they line up like this, and why, you have to have a PhD and above my pay grade to really understand how these things work. But it takes dozens of other proteins and enzymes to actually get these microtubules to form, okay, and to control when you're forming them. Okay? So there's signalers and receptors and all sorts of things. So it's a lot more complicated than this. But when you see it under the electron microscope, you can actually see the tubes. They line up in threes like that. Okay? So they really do look like that. But how they function is still, well, not only to me, but it's not real certain how these things work. At one time, it was thought they had their own DNA, and they might be a bacterial origin, just like the mitochondria. So, but they're not. But they make microtubules, which are associated with the spindle apparatus during mitosis, remember? That's what's pulling the chromosomes apart. Okay. They also make all the microtubules that are just shooting out all over, just giving the cell structure and a certain rigidity. Okay. And basically, this says what I just said. It's important for the mitosis, and microtubules stretching out around the cell. You can imagine when you saw those little motor proteins, remember? Well, they think that that's how the cell causes the cleavage furrow. They think that you've got proteins, fibrous proteins that are stretching across the cell, and then you've got motor proteins that are pulling the sides in and sort of forming a purse knot, which then seals off to form the new cell. Because remember, these membranes don't just form cleavage furrows for no reason. They have, something's doing it, okay?
That's cool.
Did you let her
Locked out.
locked out? And one of the interesting things also is that these microtubules form the basis of those little wavy cilia, okay? And the flagella of bacteria and the flagella of sperm, okay? Those are all microtubules that are being pulled on by muscle proteins, okay? Those little active proteins, motor proteins, I should say, not muscle. Okay? That's how you get the motion of these things. Yep.
I was just going to, so the cilia, the little hairs, are actually microtubules
Right.
that are just being pulled on by motor proteins.
Right. It takes ATP to run it. But if you align them in the right way, and I can't really give you the exact way that they're structured, but if you align them in the right way and then start pulling on it, it starts to spin. Okay, and that's what causes the cilia to move. That's what causes the sperm tail to move. That's what's going on. Okay, there's no muscle activity. It's just those little motor proteins doing it. But you have to have, at least in mammals and us, and in most things, you have to have the centrioles to generate that flagella or that cilia to make it.
Okay.
And there was an interesting study here where they did, I think they did laser ablation of the flies in the early cells, and they knocked out the centrosome, the centrioles. But apparently they could still generate the microtubules. I don't think you're going to see that in humans. You're certainly not going to see an ethical experiment doing that. Okay, anyway. And one of the interesting things that's now new in medicine since I was in medical school is a class of diseases called ciliopathies. And these things, every cell, we know we originated from a cell that had a flagella or a cilia, the microorganism, because that's how we got the structures. Okay? And our baseline ovum, and when they first get together and form that little blastocyst, which is half a dozen cells and that sort of thing, you have to have cilia moving on the outside of these things that are moving chemicals around and causing chemical gradients, which are actually signals for the egg to know what's front and what's back, what's side and what's right and left. That's how the body grows from head to tail, because there's a chemical gradient that's signaling genes with a different concentration. And that's how we develop bodies the way we do. And all you got to do is change that around a little bit, and you'll develop bodies that grow differently. You can, if you get a really weird sort of thing, you can grow backwards. You can grow the mirror image. Okay, this is what looks like a normal chest X-ray switched. Okay, because the heart's on the right side here. The heartbeat's not going to be here over the left. It's going to be here over the right. Okay, and I've seen a couple of these patients. I actually diagnosed one. He just came in with a cough or something, and I was listening. I got a chest X-ray. Sure enough, his heart was on the other side. But this is called situs inversus, when you see this. And it can happen not only to the chest and heart. If it's full-fledged situs inversus, you've got a mirror imaging everywhere in the body. The liver is on the left, the stomach's on the right, the appendix is not on the right, it's over on the left. Everything is switched over. It's a mirror image. Totally normal, developed normal, but it's just a switch over. Okay? And it's
And that would have been a problem because the centrioles weren't directing where everything was supposed to be going.
Right. And it happens a lot in twins for some reason, where you get one twin, and they think it's because the cilia of one twin was bothering the other one and screwed it up, and made it develop opposite. Okay. So that's, stop touching me, stop touching me.
Started early on.
Started early on. Don't cross that line. But also the cells need these signals, these compounds, the chemical signals that are being moved around by the cilia. The cells seem to need this to know how to line up. You know, when we looked in the kidney and you saw that simple cuboidal epithelium forming those tubules, well, how do the cells know to make lines? Okay, it's because of these cell signaling processes, and it takes the cilia to do it. At least for this type of structure to form. And so when you have a problem, a ciliopathy, those tubules don't form right, and you get all wild, convoluted tubes, and you basically get cysts instead of tubes. Okay, so this kind of disease, we now know that cystic kidney disease, polycystic ovary disease, polycystic liver disease, a lot of these cystic disorders are now all related, and they're all ciliopathies related to centriole problems. Okay. There's one called Meckel-Gruber that's common in Finland, because the Finns are kind of an inbred group. Okay. I read once that 75% of the Finns that live there in the country now are all related genetically to one guy who lived about 3,000 years ago. Okay, so it's a fairly inbred group, and that's where you tend to start seeing these rare disorders, the autosomal recessive disorders. Okay, the Ashkenazi Jews, the French Canadians who then went down to Louisiana and were Cajuns. Okay, these are all inbred groups that didn't have a lot of extra genes coming in. So these autosomal diseases become more and more common. So the Finns have a fairly high incidence of this, where you don't see it much in the world at all, anywhere. 1 per 10,000. So there's going to be, 50 of them in Polk County if this was the case. Okay, if it was the case here. Okay, but they have dysplastic kidneys, which tend to be cystic, and they get huge, and they have an occipital encephalocele is very common. Polydactyly just means more than 5 fingers or toes. Okay, and I guess I should say that they also know that it's a chemical gradient across the limb buds that signals what genes to come on, whether you're going to make a finger or not. Okay, so if the cilia is not active, you may not get any fingers. If it's too active, you might get 6 or 7 fingers. Okay, or it just might be the opposite of what I just said. I'm not real sure, but it's a cilia problem when you tend to get extra limbs growing. I mean, some people will grow an extra limb out their armpit. Okay, it's not a twin; it's a real extra limb. Okay, and these are this type of problem. So.
What about webbed feet?
Well, that's the same kind of thing, but what happens there is not that you didn't grow the right number of fingers. There's a signal called apoptosis that those cells are programmed to die. Okay, so when you're born or when you're a fetus, your fingers are actually fused together with skin, and then the genes turn on in between the fingers and basically tell themselves to kill themselves. The lysosomes rupture and kill the cell, and the tissues form the fingers. That's how you get rid of the webs before you're born. Okay? So they're actually normal cells that are programmed to die. So then they separate.
But didn't die.
But they didn't die for some reason. Yeah, they didn't get the signal. Okay?
Now, growing up, I had a friend who had a normal thumb, but all the other fingers were quite short. You know, not normal in growth. Would that have been like a cilia? I know hard to say.
Well, not necessarily, because you also have to have the genes that cause the cascade of development. So one of those genes could have been bad at some point in the cascade, and it doesn't matter what kind of signal you give it, it wasn't going to work. So there's a various ways to get to the same problem. Okay. But here, I think we've talked about how autosomal recessive disorders are spread. I'm sure we need to do this again. And this is PBS, gang. We're not doing genetics. You get that in AFP too. Okay. But just in case you're curious again, if I didn't talk about this, let's say this is something like sickle cell. Okay. You've got 2 carriers, parents. Each one, say the little r is the bad gene. Okay. They got one good one from his mother or father and one bad one from his mother or father. So he became a carrier. She became a carrier the same way. Now, when they get together, if they had 4 kids, they've got a 1 in 4 chance that both bad genes will get together in the same child, okay, and cause sickle cell disease. Okay? This is how all the autosomal recessive disorders work, whether it's Tay-Sachs or a lot of those storage disorders or a lot of the neurological disorders that the Amish have because they're so inbred. Okay? Carriers give a 1 in 4 chance of having a child with the disease, and you'll have 2 children on average that are carriers themselves, but then you also have the chance that one child might get a normal and not have anything, he's not a carrier, he doesn't have the disease, so you're back to normal. Okay. But this is what the dysplastic kidney looks like in the Meffel-Gruber syndrome. This is normal. If you sliced open that fetal kidney, you'd see the urine collecting ducts here. This is the area where you were looking at under the microscope where you saw the tubules, okay? And they all run urine into this area, and then it runs out through the ureter. But look at the structure here. If you can't make these tubes, you're going to be making urine and fluid, and all it's going to do is back up and start to cause little balloons, and so it destroys the kidney. Okay, and it gets huge. I mean, they can be the size of a football in a newborn. Okay, so, but that's a bad disorder. It doesn't work. You need dialysis or kidney transplant. Here's an encephalocele. Usually this is just full of fluid, cerebrospinal fluid, but obviously you deliver this by a C-section. Okay.
They don't live very long.
Not necessarily. Sometimes you can surgically just take it off. If it's just full of fluid, you can surgically remove it. Okay. It depends on how big the encephalocele is, the opening in the back of the head, and how much connection there is with brain tissue and that sort of thing. If you're lucky, it's just like having a balloon on the back of your head. Okay. But that's called an encephalocele. Okay. An omphalocele is when the same thing happens with the umbilicus, and all your intestines are outside in a big bag. Okay. And they usually have to push all that back in
But
there and surgically take it off. And this is polydactyly. The guy's got 6 fingers. It's not Photoshop. That's a real hand. That's a real X-ray. Okay. For those of you who are in lab this afternoon, what's the carpal bone right next to the thumb there?
Is that the
Trapezium, yeah. The thumb glides easy on the trapeze. Okay. The rest of you will get all these carpal bones tonight, and we'll go over those. Okay. All right, now we're just back to our organelles. The last one that I really want to talk about here is the ribosome. Very interesting, very old organelle. It's if you can think about where life almost really had to begin, you can almost look at a ribosome, because we now know that DNA wasn't stable enough to be the original code molecule. RNA is much more stable. The ribosomes are made out of RNA, which is copied from the DNA into RNA, and then the nucleoli, remember, are packaging this RNA into these pockets like this, which it basically eventually gets this sort of wrapping of this blue protein on it. But the actual organelle itself is mostly RNA. Okay, this is one of the subunits. There's another unit that comes in and joins it to form the active ribosome. Okay? But you can see it looks like a single strand of DNA right here. It's not the double strand, it's just a single strand, okay, and then it folds up on itself, and those are much more stable than DNA is. Okay? And they think that the first self-replicating molecules were RNA molecules, and we know that to make protein you need a ribosome, so you need RNA. Okay? This was the only, let's call it an enzyme. This was the only RNA structure that actually worked as an enzyme that anybody knew about for years, until the early 1990s when they started finding other enzymes in the body where RNA was actually doing the work. Okay, so and now it's over 1,000 of them have been found just in the past 20 years. Okay, so now we think that it was an RNA world that first got things going, and then DNA had to somehow be a side offshoot of the RNA. In other words, for some reason, the DNA was a copy of the RNA, the original RNA. Okay.
But now the RNA copies the DNA?
Now the DNA is copied to make RNA.
To make RNA.
But we think it had to start out like RNA. Okay, it's much more stable. So, but it's basically found in all cells because all cells need protein. So and you need a ribosome to make protein. Okay, so this is if this is about as close to the beginning of life that you can think about. They've actually found RNA that makes copies of itself. And by any stretch, that's the definition of life, when you're replicating and reproducing. Okay, so they think that that's what was going on. So, okay. Now we'll go hit the new slide. I sent them out. The first few things here are going to be terribly important. Just take a few notes. It's relatively simple here. Okay. So, we talked about the structure of cells. We talked about the organelles and how they function and what they do. And then your book goes into this discussion of, well, now you're going to put together multicellular organisms. And a lot of other single-cell animals and plants have all those other organelles that working for them. But now you're going to build something. So what holds these things together? Well, it took a couple of billion years for these things to start developing these junction-type proteins here. Obviously, because they're keratin and actin and all sorts of things, these sorts of proteins were inside the cell doing other things, but at some point the proteins developed a way to hook onto each other to form multicellular organisms, and that had to happen. And there are several different types of things, and I'll show you some individual pictures of these as we go on in the next few slides. But so it's just, these are all the different ways that the cells can adhere to each other. So some of them will be called cadherins, like we talked about before. But this is the basement membrane. Let's say this is the lining, the simple columnar lining in your GI tract. Okay, there's the basement membrane along all epithelial linings. This is what they stick to, so that they don't just get washed off as the food goes by. But they also got to be watertight between them. Okay, you don't want all the stuff in your intestines or your stomach to be seeping between the cells. Okay, so you have to form these watertight bonds between the epithelial linings. Okay, and this is just some of the ways they found to do it. There are belts that go all the way around the cell, and it's a sort of a strap like this. You have those actin fibers that run around the cell, and then you have the cadherin molecules and others that will grab onto each other just like Velcro. But on a molecular level, that becomes waterproof, and it holds these things together. So this could be gastric acid out here, and it can't get between the cells to destroy the basement membrane and do anything else below it. Okay? So, but a really interesting structure here, the way these things wrap around on the proteins to hold and give it strength. And a fair amount is known about all these things now. So, and this was just imagined when I was in medical school. So in the last 30 years, there's a lot of new stuff going on. One little PBS moment I think you might find interesting. It's thought that, this actin, those fibers, that's the same fiber that the little motor proteins are walking on in your muscles. Okay? And we'll talk a lot about that when we get there. But it's thought that these fibers, arranged kind of like this, were or are the way that cells will change shape. The embryo, you've all seen pictures of human embryos, just a big ball of cells. Well, eventually it becomes a ball of hollow cells, okay? But then it invaginates. Then it has part of its wall moves in and opens up, okay? And that's what forms the GI tract, the mouth and anus, okay? And so it sort of invaginates and forms that opening. It's like I've got a balloon and I'm just pushing my fist through it, okay? But these cells don't have minds. They don't know where to go. They don't, what's causing this? Well, it's chemical signals, obviously, forming like I said, forming the nose and tail end and that sort of thing. But it's thought that the actual movement, the invagination, is caused by motor proteins pulling on these actin fibers. So if you can imagine the cell looks like this, and the actin fibers are being pulled in, well, the next one being pulled in, what are you going to get? You're going to get a curvature. Okay, so you're eventually going to get that thing to move in. Okay, so it's thought that that's how that happens. Okay. Okay. Tight junctions, you can just look at them as rivets. They're just protein rivets. Okay, and they'll form rows as well. Okay, and so that's going to be, again, another waterproof barrier for things that can't get between the cells here. Okay, what do I want you to know? Just remember that tight junctions hold cells together, and they're really useful in epithelial linings. Okay, because you want them to be weatherproof, basically. Okay, don't worry about the names here. Okay, here's an actual picture of one. This is an endothelial cell of a capillary. Okay. The blood cell would almost be the same size as the capillary. Okay. Now, these really small capillaries are only one cell wide. Okay. And they wrap around and grab their tails with tight junctions. Okay. That's how they seal themselves in the end, in the tail. The other interesting thing about this is that we're going to look at the processes of endocytosis and exocytosis, where cells bring things in through the membrane and then release things through the membrane. This is what's going on right here. These are the vesicles right here on the inside. They're picking up nutrients and all sorts of chemicals that they want. They may have receptors that will grab them, and then the vesicle forms around it and comes in. They're dragged over to the other side, usually by microtubules. Okay. They have docking proteins over here, so the vesicles know that this is the outside, this is where I'm supposed to go, okay? This is where I fit. And then you have special proteins that are calcium triggered that open up and release the stuff to the other side. It's fantastically complicated. It's not just these things banging into each other. But these are the endocytosis vesicles here, and then these are the ones that are going to let the stuff out into the surrounding fluids on the outside of the capillary. So these are the ones that are going to feed the cells that are living here. Okay. So, but interesting little setup there. And remember, the lining, the simple squamous lining of capillaries, those are, it's called the endothelium. Okay. Gap junctions are another way that cells hook up together. This is actually a very interesting and an absolutely necessary thing for your smooth muscle and your heart to work. Okay? I'm not quite sure how many other cells have a lot of gap junctions, but I know that the heart muscle, the cardiac muscle, and the smooth muscle, say in your intestines, have a lot of these things. And it's just a protein that the cell has put in the membrane, which bonds to the same type of protein in the cell membrane of the next cell. And it creates a gap. That's why they call it a gap junction. So you can get all sorts of things flowing through here, okay? Calcium ions, sodium, potassium, whatever. So this cell can communicate with this cell, okay? And in muscle cells, calcium is the trigger for contraction, okay? So this is how you get rhythmic contractions of, say, the heart muscle, or rhythmic contractions, the peristalsis in the intestines, okay? What you've got is cells that you only have to have a nerve that fires and gets one cell to contract, okay? It releases calcium inside itself, okay, to make all the motor proteins fire and contract. But that calcium can get through the gap junction and cause the next cell to contract. So that causes this wave as you go. And so you get this rhythmic beat of the heart instead of everybody having to be stimulated in order, okay? So you get an automatic rhythm because of these gap junctions. Okay? In the same way with contractions in the uterus during delivery, okay? That's what happens. A lot of gap junctions because that's all smooth muscle as well. So, okay. Here's an actual shot of a bunch of gap junctions. You can actually see the round protein in the hole. Okay. So you can imagine it's like almost the whole side of this membrane is just full of them, so calcium or whatever is just going to be swishing through here, okay? So that's an interesting picture. And believe me, never saw anything like this when I was in school. So, desmosomes. This is more sort of a mundane way of just hooking cells together. Again, you'll notice it's keratin. These are mostly in the epithelium of the skin, okay? Got these big proteins here, and then you've got a membrane conglomeration of other proteins with others that have receptors for each other that will join, mainly hydrogen bonding. I don't think any of this is covalent bonding here. But it forms this real weird kind of fuzzy plug that just, again, like a rivet, that just holds the cells together. But then if you can think of, like it is up here, okay? But then if you can think of a half of this and just flip this over and stick it to the basement membrane. That's how they stick to the basement membrane, is when a lot of these, and when it's only a half, they call it a hemi or a demi-desmosome, okay? Here's an actual shot of one. You can see how the membrane gets much thicker with all the proteins there. And then you have these keratin fibers flowing throughout the cell this way. But that's where it's stuck, right there in the center. Those are the two membranes.
Dr. Erks,
Yeah.
what kind of cell would that be?
What kind of cell?
Yeah, are there specific cells that wind up using a
Yeah, well, what cells do you know that make keratin?
Oh, yeah. Okay.
Epithelium.
Okay.
So, okay. So here, when you have those hemidesmosomes that are attached to the basement membrane, everybody now recognize this tissue, right? Keratin. This is stratified squamous. This is actually probably a dark-skinned person. Those are the melanocytes in there. But the basement membrane has been destroyed. It's no longer connected to the irregular connective tissue down below. And this fills up with fluid, and that's the definition of a blister. Okay? So that's what this is over here. Their whole epithelium is just lifting off and filling up with fluid underneath.
It says it's not a genetic disorder.
Right, right.
So what?
This is one that like I say, I've seen 10, 15 cases maybe of this. This is a drug reaction usually antibiotics, real common with penicillin type drugs. It triggers the immune system somehow to make antibodies against your basement membrane, against those desmosomes, and it destroys the connection so that it just lifts up. Okay, and it usually lasts about 6 weeks or so. You just put them on a little steroid.
Or to grow away? It's
sorry?
reversible?
Yeah, it's not fatal.
Is that an allergic reaction to?
No. Well, not technically, but yeah, it's a drug reaction. But at least when I think of allergic reactions, I think of histamine release, that sort of thing. This actually takes a little while. You can be on the drug for several weeks, and all of a sudden it fires off because it has to trigger the immune system to do this wacky thing it doesn't want to do, which is destroy your epithelial basement membrane. Okay, so but it usually takes about 6 weeks, and then you recover. The blisters heal up, and you look like you've been burned, but you're alive and you're fine. Okay, but it's obviously you got to worry about infections and all sorts of things here.
So would a mild case of that be like if your hands peel or
Yeah.
something? Okay.
Yeah. You can get drug reactions where you lose all sorts of things. Your fingernails, your hair, the skin on the palm of your hands. But very often, that's more the disease than the drug. Okay, there's a lot of viral diseases that will cause your skin to peel. Okay,
So when you're treated with steroids, then it reattaches to the basement membrane? Or?
no, it won't. It has nothing to do with reattachment. When you give anybody steroids, all you're doing is turning down their immune system. Okay, so you're just kind of turning down the volume on those antibodies, but you're not really affecting what's going on as far as the rebuilding of the structure.
Oh, so, but it eventually does reattach.
Yeah. Because your skin grows pretty quick. And all these cells are growing, and they're trying to make desmosomes. And, so it's a battle for a few weeks, but you eventually win this usually. Okay.
Can I ask you a question that's kind of related, but
Sure.
maybe not?
Please.
I had a one time where I was working with butternut squash and it was raw, and all of a sudden my hand just started to kind of burn, and the skin just kept peeling off. And apparently it's a very common thing because of the starches in the
With butternut squash?
with butternut squash. Yeah, I know it sounds really bizarre, and never had it happen before. But I just happened to peel it, and I was chopping it instead of cooking it first. And I did some research, and I mean, in a few days, your skin obviously heals
Yeah,
itself.
yeah.
But literally, it's like you could barely see the fingerprints because the skin just kept peeling, and it would burn.
It's like a second-degree burn, yeah.
It was,
You're
and
sloughing the epidermis.
yeah, and apparently it's fairly common. It's not an uncommon thing to have happen.
I've never heard of butternut squash being an issue.
Yeah, and I know it sounds really, really weird. You can, I mean, I was freaked out, obviously, because, you know.
I mean, obviously there are a lot of chemicals and a lot of foods that can do that.
Mm-hmm.
I didn't know that butternut was common.
Yeah, I did a,
I had a similar reaction with figs. Real
Really?
figs off a
Yeah.
fig tree.
yeah,
When I was about 12 years old, we were pulling them off and throwing them at each other, and I got hit in the forehead with one. And the white milky stuff,
Yeah.
you know, like almost like milkweed.
yeah.
It's,
Figs have,
I didn't think anything about it, just wipe it off, and then I'm here, take this.
yeah.
But the next day, my face was one big blister.
Oh, wow.
One of it's the starches.
And, well, not just starches, but the
Chemicals.
chemicals.
The chemicals, okay, so
There-
it's not
There's
just
some
the,
reactive chemicals in those things, and you never know what you're going to react to. And some people will do it with strawberries, others, you know.
Can you develop an allergy? Like I used to
New one, yeah.
Like I used to eat strawberries all the time when I was little, and now anytime I eat them, my tongue automatically swells up.
Yeah, yeah. Yeah, you can develop, especially to
I
foods.
thought you
and
were
things
supposed to
like
grow out of allergies.
that. Well, you can do that too. Both ways. You can get new allergies and you can get rid of old ones. But you can't always predict. Half the people who think they're allergic to penicillin aren't anymore. Luckily, because when they get it by mistake, they don't die. So that saved a few residents that I knew.
I have a question. Have you ever heard of like the melting people?
Melting
Like
people.
their face looks like it's kind of melting. Would
With
that
the
be
skin
part of
disorder?
the- Would it be like that? Maybe? I watch weird stuff, so I'm sorry.
Just from what you're saying, if the
It's like
skin
their
is sagging,
skin is like really, it looks like they're melting.
yeah, I would blame that on some sort of collagen
Collagen.
disorder or elastin disorder where the skin just has no elasticity.
Yeah, like I mean, it was a lady.
But they're
It's
not
like
blistering, this is blistering.
There was one that he was blistered all over. It looked like it, or boils or something all over. Would that be part of that?
I can't even tell what the disease you're
It
talking
was like
about.
on the taboo or something.
Well, I'll maybe do a little search for droopy skin disease.
It's like the
Melting
tree, it was like
people.
the tree guy.
I'll see what I can come up with and see
Well,
what's
the
the
tree
basis.
guy that you showed us,
Yeah.
it was some of his friends or whatever, like they went around in a circus, like
Oh,
The
weird.
tree in there now.
so it's a real
It's a, yeah, I mean,
I'll look it up, see what's.
yeah, I'm sorry.
But I'll bet it's similar to the guy who could take his skin, pull it out.
But
It's
this, I mean,
probably
her
the same.
eyeball was like her eye, it's part was
Yeah.
down here. And,
I'll look into it, melting people. Okay, well, good, I like that. Is this related to this, or I had a friend that had this, and this is where you all learn something, okay? You're gonna remember that more than you will desmosome, right?